Anyway, passive and active transport for the umpteen hundred times. So passive transport, this is where we are not using energy. This is where things are just diffusing from high to low. The only time that we have something that's not spreading out and diffusing is osmosis, where it's going to be attracted directly towards salt or higher salt concentration in the solution. So our phospholipid bilayer is made up of hydrophilic heads and hydrophobic tails. So this head right here is going to be attracted to water, and hydrophobic just means it's going to be repelled by water, so the tails will flip inwards away from water and cytoplasm. And it forms a bilayer or a phospholipid bilayer. That's actually what our first one down here is. So phospholipid bilayer is this because it has two layers connected. Osmosis is going to always deal with water, and that's going to be it being attracted to salt. Simple diffusion is going to be whenever it goes straight through the membrane, so oxygen goes straight to the membrane. Facilitated diffusion is when it goes through a protein channel or transport protein like glucose. Since glucose is a bigger molecule, it has to go through a channel protein, where oxygen, which is a smaller molecule, can just go straight through it. Water usually uses an aquaporin, or it just goes straight through the membrane. And since it's repelled, it just goes straight through. Once again, based on salt concentrations. So, on this area, once again, if we have water leaving the cell, there has to be more salt outside the cell. Hypertonic has the high concentration of salt. All of these are examples of passive transport because we're not using ATP and it's not forming vesicles, which is also using energy. Our membranes are not impermeable, they're semi-permeable. They can kind of be gone through. So once again, things are sneaking through one direction or the other. The glucose is going inside the cell, which means that as a result, there has to be more concentration outside the cell. Over time, our cell is going to shrink because because water is leaving the cell, and oxygen does not require a channel protein to move through the membrane. So on active transport, this is the ones that require energy. This can be in the form of ATP. What we need to know here is this is steps 1, 2, 3, 4. We are wanting an electrochemical gradient, so we have more positive charges on one end than the other, on the outside of our cell than on the inside. This just means that we're pushing ions on the other side. And then here are the steps for this process. Please pause this and go ahead and grab this info. And this is just called the sodium-potassium ion pump. I'm going to use Na for sodium because it was once called natrium, and K for potassium because it was once called kalium. So with this, what's happening is sodium will bind to this pump. ATP will then bind to it as well and release the sodium on the other side. Potassium comes in. The ADP, which is adenosine diphosphate, because now it has one less phosphate group, will then break off, and the phosphate group and the potassium is released back inside the cell. On the other end of thing is vesicle formation. This is endocytosis and exocytosis. Those words seem scary. Please pause conversation. These words seem scary, but all they are is importing into the cell and exporting exporting out of the cell. So we just are bringing in solid food particles, bringing in extracellular fluid, add these arrows, or due to receptors, we're bringing in some kind of protein or hormone. Last big thing is exocytosis. It's exporting. We see exo, we know it's exporting. This is, once again, vesicle formation. If we form vesicles like these, once again, these bubbles of phospholipids, it is going to require energy. It's going to be active transport. If we use ATP or we see vesicle formation, it's active transport. And once again, this is for proteins, neurotransmitters, and waste. We can also take in neurotransmitters and proteins, hopefully not taking in waste, through receptor-mediated endocytosis. And that's basically all this is. Y'all got this. Ask questions.